Welcome to the AI Research Roundup. I'm Alex, and today we're looking at a paper from the Hugging Face trending list, published on July 2nd, 2025, just 2 days ago. It introduces a new biomolecular structure prediction model that achieves accuracy comparable to the state-of-the-art AlphaFold 3. The paper is titled IntFold, a controllable foundation model for general and specialized biomolecular structure prediction. And its ability to be specialized for specific drug discovery tasks is what really makes it stand out. This paper shows how small, trainable modules can adapt a massive base model for highly specific and challenging predictions. Also, the authors shared their implementation on GitHub. It looks pretty comprehensive. You'll find the full source code, of course, but also quick start commands to get it running with pip. They've included installation guides and examples to help you start predicting your own structures right away. Alright, so let's look at how the model actually works. This diagram shows the IntFold architecture. It starts with an input sequence, like a protein or DNA, and feeds it into the main model. The key innovation here is this small adapter module. An adapter is a lightweight, trainable component that lets researchers specialize the model's behavior without retraining the whole thing. The model then generates structures, which are ranked to get a final prediction. The bottom panel shows how these adapters enable specific tasks, like guided folding, or even predicting binding affinity. Building on that architecture, these charts show us the results, and they are impressive. For predicting single proteins, IntFold achieves a mean LDDT score of 0.88. LDDT, or the Local Distance Difference Test, is a score measuring the local accuracy of a predicted protein structure, and that 0.88 is right on par with AlphaFold 3. It also matches AlphaFold 3 on protein-protein interactions. But for really tough antibody-antigen interfaces, the specialized IntFold Plus model significantly closes the performance gap, showing the power of those adapters we talked about. So this is where that specialization really shines. This figure focuses on a protein called CDK2, which is important in drug discovery. It has a main site and a second allosteric pocket. Allostery is when binding at one spot changes the protein's shape somewhere else, a really subtle but critical effect. The general model, shown in green, struggles to predict this with high error values. The number you see is the LRMSD, or ligand root mean square deviation, a measure of error, so lower is better. Notice how the fine-tuned model gets incredibly close. And this idea of guiding the model goes even further. These charts show what happens when you give the model a small hint, called a structural constraint. For tricky antibody-antigen interfaces, the success rate nearly doubles, jumping from about 37% to 69%. The image on the right makes this really clear. The initial prediction is way off, with a DocQ score of just 0.06. A DocQ score measures how well proteins are docked from 0 to 1. After adding one constraint, the prediction becomes nearly perfect. Okay, so beyond just predicting the shape of an interaction, the model can also predict its strength, a crucial factor called binding affinity. The bar charts on the left show how IntFold stacks up against other methods. The score here is AUPR, which is a performance metric where higher values are better, and IntFold clearly comes out on top. Then, the scatter plots on the right show how well the predicted affinity matches real lab experiments. The Pearson correlation coefficient, or PCC, measures this relationship, and again, IntFold's predictions show a stronger correlation with reality. Okay, so this table gives us a broader look at the model's general performance. It's a head-to-head comparison between IntFold and another model, Boltz-2, on a really recent test set, which is important for a fair evaluation. Looking at the results for predicting single protein molecules, or monomers, they're basically tied. But for predicting the interfaces, how different molecules connect, IntFold consistently comes out ahead. The most dramatic difference is in antibody-antigen prediction, where IntFold's success rate is over 15 percentage points higher. This really underscores its strength as a powerful general-purpose prediction tool. So to wrap things up, IntFold really establishes itself as a state-of-the-art model, matching the performance of giants like AlphaFold 3. But its true innovation is that adaptability. Through those lightweight adapter modules, which are small, trainable add-ons, it can be fine-tuned for very specific, difficult tasks in drug discovery, like predicting subtle allosteric states. This combination of raw power and precise control is what makes it so promising. That's all for this week's AI Research Roundup. Thanks for tuning in.